Good evening, everybody, and welcome to the fourth public lecture of the Ocean Institute for 2024. My name is Christophe Godin. I'm the director of the Ocean Institute here at UWA, and it's my pleasure to welcome you tonight and to MC this event. I'd like to start by acknowledging that the UWA campus is situated on Noongar land, and that Noongar people remain the spiritual and cultural custodian of our land, and continue to practice their values, languages, beliefs, and knowledge. I just have a very quick forward before we get to business. So as I mentioned, this is the fourth lecture of the UI this year. This is part of a new thinking we've been having with the UI executive team, over the year that led to a new strategy that was launched in March this year. And that reflected on the role and the mandate of an institute like us, in public engagement. And it was very evident to us, as we were progressing our thinking, that the awareness of UI activities and the weight it has in the public debates and the definition of our research outcomes is not commensurate to actually the quality of the research activities that we are undertaking. And so these public lectures are part of a new series of events that we'll have organized and that we'll keep organizing to try to actually reach out to a much broader audience. This time we are doing it in the UWA campus, but in the past we've been going to the city and to Fremantle, to the museum and to other locations, to actually reach a broader audience, and raise the awareness of not only the research activities and research outcomes we are producing, but also the main challenges that the Ocean Institute is trying to address at the moment, and essentially the climate change crisis and the biodiversity crisis. So it will be more next year. We'll continue that series, and so please stay in touch and follow the newsletters, and if you are in the mailing list, there will be one in the first quarter next year, and we'll be continuing this series of public lecture in 2025, and along with other events that we are planning to organize. This lecture is actually quite special for us because it's the lecture delivered by the inaugural chair of the Jock Clough Marine Foundation, which is something very special. And this is the opportunity for us to reflect on the importance of philanthropic donation to research these days in universities. As you know, reading the news left and right, universities are under significant pressure across the board. Research funding is decreasing quite significantly. And there is actually a disconnect between the expectation of the general public to have science totally independent, and at the same time, a lack of independent and government funding to actually undertake this research. And in this new context and in this new landscape, we rely more and more on industry funding, which has benefit, but also creates some challenges, but also on philanthropic funding. And the Jock Clough Marine Foundation, with the support of Jock Clough, has been absolutely fundamental in the activities of the UI for nearly a decade now. But I think something which I want to reflect very specifically coming from Jock is that it's not only the funding that has been important, it's actually Jock's engagement into all the activities of the Ocean Institute, his ability to actually network and disseminate our activities, the mentoring he's been providing to me and to previous directors, and as well to researchers and so on, and the continuous belief of that what we are doing is actually important for our community. So I want to take this opportunity tonight, Jock, to thank you and to express my gratitude for your engagement and your support of the Ocean Institute. So thank you very much. It's enough for me tonight, and so now I've got the pleasure to introduce Dr. Charlotte Birkmanis. Charlotte is a scientist and executive bridging the gap between research, philanthropy, and the public. She is a marine biologist and explorer trained in ecology, researching predator and ecosystem conservation and management with a focus on shark, fish, and marine megafauna. Charlotte is an Australian STEM ambassador, author, and podcaster, and most importantly, an adjunct research fellow at the Ocean Institute. So please welcome Charlotte.
Thank you, Christophe. Just move these along. Thank you for that wonderful introduction. I'm also the director of the Jock Clough Marine Foundation, and I'd also like to echo Christophe's words and thank Jock for his mentoring of me personally, and also for everything that he does for the university and for the foundation. Now on to the main event. We have Professor Steve Swearer, who is the Jock, Clough Marine Foundation Oceans Chair. And I've also had the pleasure of working with him through the foundation, and he works with us. And I've also been inspired by the ideas that he has and the work that he brings with him. And I think he's going to give an overview of some of that today. And his interest spans the early life histories of fishes to the ecological responses of coastal marine ecosystems to environmental change. And his efforts are currently focused on partnerships with government and industry to provide practical solutions for improving the sustainable management of coastal marine resources. So that's enough from me as well, and please welcome Steve.
Okay, thank you very much. I do work on a variety of different things, and I needed to simplify it and have a focus topic that I could cover today. And the decision was that I would spend a little bit of time talking to you a little bit about the marine ecosystem restoration work that I've been, and my group have been doing over the last 10 to 15 years. And so I'm going to take you on a bit of a journey around the kind of what, how, and why of marine ecosystem restoration. And I'll give you a couple kind of case studies to illustrate, I guess, some of my perspectives on where I think we are and where we need to go. So the current geological age has been coined the Anthropocene, and this is to capture the rapid acceleration of human impact on planet Earth. And one of the consequences of this is that as societies have evolved and grown, is that we've replaced natural habitats with alternative uses, such as farming and cities, and this has led to global scales of habitat loss across many and most terrestrial biomes. But that change and impact doesn't stop at the coastline. These impacts have also been more recently recognized, because it's a bit harder to see underwater, losses of coastal marine ecosystems. And there's a range of estimates about how much we've lost. Some of this is hard to do because our historical baselines have shifted, but our estimates are that we've lost somewhere on the order of about 85% of historical levels of coastal shellfish reefs. We've lost approximately about 50% of global coral reefs over the last 50 years, about 35% of mangrove forests over a comparative amount of time. For salt marshes, this is difficult because we've lost a lot of them and we don't have good baselines, but we know that we've lost at least about 6% in just the last 20 years. We've lost about 30% of global seagrass cover, and we know we have at least reasonable estimates that currently we're losing kelp forests at a rate of about 2% per year. This is not good, and we need to garner and incentivize people to actually engage in trying to bring these habitats back through restoration activities. What I'm going to show you here is the results of a study done by Megan Saunders at CSIRO and colleagues that effectively did a literature review that really encapsulates the kind of timeline of marine habitat restoration relative to terrestrial forest restoration. And what we can see on this plot on the left, or in figure A, is we've got kind of 4 groupings of when there is the first kind of anecdotal story published in the literature around a habitat restoration project. And the symbols for both plots, if you want to figure out what ecosystem they are, that's in the upper left-hand corner of panel B. So what you can see is that they found evidence that, at least anecdotally, there are old shellfish reef restoration projects that date back to the late 1800s. But what you can see is the pluses represent the timelines for the earliest anecdotal story in the literature versus the first publication versus when the 100th publication of that particular habitat had been restored. And then the modern one is habitat restoration that followed modern ecosystem restoration principles. So effectively what you can see is that from a marine ecosystem habitat restoration perspective, it's a relatively new phenomenon. We've only really been doing this since about the the latter half of the 19th century. And if we then look at this in relation to the timeline, you can see that more clearly. But more importantly, even though there's been an uplift and an increase in the number of studies that have engaged in marine ecosystem habitat restoration, they still represent a very small proportion of studies involved more broadly in the field of conservation biology. So the take-home messages here are we've only really been doing this for a short amount of time, and we're not actually allocating a lot of effort relative to other conservation efforts. Another challenge is, how do we upscale? A lot of restoration projects, particularly in marine realms, are typically pretty small. The majority of them are only operating on a scale of about a hectare. So we're talking about 100 meters by 100 meters. But we know we can do it at scale. And so this review by Megan identified kind of bright lights of successful marine ecosystem restoration projects that have been successful at scale. So we can restore shellfish reefs at hundreds of hectares. Coral reefs are a bit more of a challenge. Large-scale successful projects for mangroves in particular. So we can do this at scale. We're going to need to be able to do this at scale if we are going to achieve global goals for nature. So we're going to be able to halt biodiversity declines and recover biodiversity through conservation and restoration practices, which is what that green curve is. But as has been argued, and I'll give some examples of this, we actually need to think differently about how we actually produce resources in a way that can be positive for nature, not just for society. And this is particularly true and relevant in relation to trying to achieve goals associated with the UN's Decade of Ecosystem Restoration. So how do we actually go about restoring these habitats? And ultimately, we need to provide answers to some relatively straightforward questions. So initially, we need to have a better understanding of what actually caused the loss of the habitat in the first place. Can recovery of that habitat happen by simply removing the stressor or the cause of that habitat loss? And if not, if we have to engage in active restoration efforts, then what interventions do we actually need to engage in? So is it a situation where the organism can't recover naturally because it's limited by available habitat? So in a marine context, this is often referred to as substrate limitation. Or is it that the organism can't get to this area that you've restored because the propagules, the spores, the seeds, the larvae cannot get there because source populations are too far away? Or is it that the environment's simply become unsuitable? So have things happen in the environment, such as global warming, that mean that species may not be able to survive in that particular location where they used to historically have been capable of persisting. So to give you some examples of how people are tackling some of these challenges, this is some work that's being done by an NGO in Indonesia, where the challenge here is that a lot of coral loss in this system has actually been through physical damage, and that results in very large fields of coral rubble. And this coral rubble is very dynamic. It's sort of like a constantly shifting substrate, and any coral propagules that are actually settling into this mosaic actually don't survive. And so the solution here is the availability of stable substrates, and it's about how you create stable substrates to allow coral recovery in this system. The Seeds for Snapper program that Gary and others are heavily involved in is an example of a propagule limited system where they're actually putting seeds of seagrasses in areas where recovery without propagule supply is unlikely to happen. And the Nature Conservancy's Reef Builder program is an example of trying to recover a lost ecosystem where we have both substrate limitation and propagule limitation supply. So TNC has been engaged in having to actually provide rock substrates so that flight oysters can actually get off soft sediments so they don't get smothered and buried. But in some areas, these substrates can't be naturally colonized because there's not enough remnant populations of flat oysters to provide spat for natural recruitment. And so TNC have partnered with aquaculture industries to actually grow flat oysters in the laboratory, settle them onto recycled scallop shells, and those scallop shells are in turn, as you can see in this photo, outplanted onto those substrates in order to recover these systems. And we might hear more about that in the panel discussion later today from Fiona. In terms of habitat suitability, there's kind of two ways that this has been addressed, and it's really around this question about how you match a phenotype of an organism with its environment. So it's a phenotype environment match mismatch. As environments change, one solution is that you change the phenotype of the organism to match how the environment is changing. And one way that is being done is using assisted evolution to identify, in this example, corals that can actually tolerate warmer ocean temperatures. The alternative is that you actually engineer or modify the environment so that you can actually maintain suitability so that habitat can be restored. And this is a problem that we've been faced with trying to recover mangrove forests in areas where the wave energy has become inhospitable for mangrove seed anchoring and seedling survival due to erosive forces. And so we basically have to mimic or reduce that mechanical energy to actually give these seeds and seedlings a helping hand. And the reason why this has happened is because the positive feedback mechanisms that an intact mangrove forest provides to early life history stages in terms of the pneumatophores, the prop roots, the tree roots that are actually reducing that energy that actually naturally provide this positive feedback, natural regeneration process have been lost. And so we have to ecologically engineer that process back to reestablish an intact mangrove stand, which can then regenerate that positive feedback mechanism. So I'm going to give you a case study of work that we've been doing with a very large team of collaborators around kelp restoration in temperate Australia, and specifically in Victoria. And the problem here is that we have overabundant urchins. And I just want to put this into context that the issue around herbivores and restoration failure is actually a fairly broad and common phenomenon. This is a recent review paper where they looked at the impact of herbivore exclusion on the outcome of, or the loss of either biomass, which is in the top, or abundance, sorry, and diversity in the bottom panel in terms of all habitats, terrestrial vegetation habitats or aquatic, so marine or freshwater habitats. The take-home message here is that, well, the green bars represent a relatively undegraded state or habitat. And so this vertical line here represents the line of no change. And so these bars that are colored bars that fall on the left-hand side indicate loss of vegetation in areas where herbivores are present relative to areas where they've been excluded. The orange bars represent planted restoration areas, and the blue bars, or kind of purpley ones, are naturally regenerated areas. What I want you to take away from this plot is that basically herbivores eat vegetation, unsurprisingly, but they eat it a lot more in a restoration plot than they do in actual natural habitats. And so you can imagine you've just provided a lot of juicy, non-chemically defended plants, for example, that can get munched. And they're also a lot easier to find than little individual soft, juicy plants in a mature forest stand, for example. So herbivore can be a major limiting factor in restoration success of vegetation habitats, specifically globally. And this is happening in Australian waters today. So this is some video shot on some reefs in Port Phillip Bay in Victoria, just showing the spatial extent of urchin grazing on some of these rocky reefs, where we're getting 30 to 50 urchins per square meter on these completely barren rocks. You can see the kind of smattering of a few sea stars, but this is completely clean rock. There is not a scrap of vegetation on it. So this is, a classic example of an urchin barren. And so we've done a fair amount of work of diving around Port Phillip to figure out, well, how many urchins do we actually have? And doing a bunch of urchin surveys and trying to determine what the relationship is between urchin abundance and macroalgal cover. And this has been found in anywhere anyone's ever looked at, unsurprisingly, the more urchins you have, the less percentage cover of macroalgae you have. And there are some thresholds, which are indicated by these large vertical white lines, that effectively at urchin densities above kind of about 8 per square meter, pretty much guaranteed to lose everything. And if you want to maintain an intact canopy or a relatively intact canopy, you need to maintain urchins at a density of around 3 to 4 per square meter, at least in this particular system. So if we want to maintain what macroalgae we have left, and we want to recover reefs that are in a urchin barren state, to recover that canopy on those reefs, we need to manage urchins. So how many urchins do we need to manage? A lot. There's about 2,200 hectares of subtidal rocky reef in Port Phillip Bay, which is actually not a lot in the grand scheme of things. And our estimates are that there's probably around 142 million urchins in the system. And if you take an average urchin size of around 80 grams, that's about 11,000 tons of urchins. That's a lot of urchins. And so many of you might be thinking, well, just harvest them, right? Well, there's people eat them, right? This is a non-problem. You're right. You would think that would be the solution. And there is a commercial fishery that got formed in kind of the late 90s, and their quota limit, their total allowable commercial catch is 60 tons per annum. The other problem is that they fish urchins that are fishable biomass, meaning they actually have something in them that's of value, and that's their roe. If you go to an urchin barren and you crack open an urchin, I can guarantee there's going to be nothing in that animal. So fishers fish in areas where urchins are not a problem. We estimate if we want to flip the system back, so reducing abundances on weeded reefs, so macroalgal dominated reefs, and reducing biomass on reefs that are barren to recover macroalgae, we need to remove about 6,000 tons of urchins, and that's about 75 million individuals. How are we going to do this? Well, in some places, this is what we're doing. They're killing them through culling. So this is actually a video of a research project done in partnership with Parks Victoria with recreational divers, actually quantifying how many urchins they were removing so they could actually document the relationship between urchin removal and kelp recovery. This is actually a different species. This is the long spine Centrostephanus in eastern Victoria. The Port Phillip Bay one is the purple urchin, which is a different species, but we get similar sorts of problems. The reason why we have to do this in marine parks and sanctuaries is that you can't commercially harvest in a marine park or sanctuary. And so the only easily management-focused solution in a park is literally culling. So we did an assessment in collaboration with colleagues at Deakin University about what's the cost-benefit of actually doing this sort of activity. And depending on where the reefs are, how hard they are to get to, how deep they are, we estimate that it's somewhere between $6,000 and $22,000 per hectare to just do the initial cull. That was just the first time of going in and smashing urchins. And roughly, given the scale of the problem in a Victorian context, that's somewhere around almost, could be upwards of about $50 million to just do the initial cull. So this is not ongoing monitoring, maintenance culling. That's all not calculated in this. If you then calculate the sort of non-market or ecosystem service benefits of rocky reefs, so whether that's the nutrient removals they do, the fishery habitat that they provide, it still actually has a benefit to cost ratio greater than 1. So you're actually getting a return that's greater than how much you've spent on doing it. But the fact of the matter is it's expensive. And trying to get people to invest that kind of money in this is a challenge. So the key point is, if you cull them, do you actually achieve the response that you're intended? Can you recover kelp by simply getting rid of the agent of loss, which is overgrazing by urchins? So in partnership with Deakin and the Nature Conservancy and Parks Victoria, we've been monitoring what happens in response to culling by commercial divers in marine parks. So this is what a whole bunch of photoquadrats look like in a pre-culling plot prior to any urchin removal. This is roughly what it starts looking like in the northern part of Port Phillip Bay after about 6 months. And that's what it looks like after about 12 months. And so what we can achieve here is that even just by removing urchins, we can restore about 25 hectares of reef in terms of at least flipping it from a completely barren state to a vegetated state simply by culling. The problem is that we're not necessarily getting back the species of macroalgae that we are intending to recover. And the project is really focused on trying to increase percentage cover of Aclonia radiata or golden kelp, which is a dominant habitat-forming seaweed in southern Australia. And so in some areas, we're not getting Aclonia back because we're propagule limited, and so we've been trialing a range of active or assisted restoration practices. So we've been using green gravel. Colleagues here at the Oceans Institute at UWA have been pioneering in developing this method. Basically the idea here is that you're taking substrates, whether they're gravel or string or twine, or even wrapping the twine around the gravel, is that you're seeding them in a lab. And so you're getting lots of little sporophytes, and then you're outplanting those fouled, if you like, substrates. And this is potentially useful in areas where the habitat may be fairly unsuitable because for example, you're getting lots of turfing algae, and that matrix that's covering the rocks is not great for spore survival. So we've tried a whole bunch of methods, including transplants, where we've taken juveniles from areas where Aclonia recruit into, but where we don't have permanent beds. So they're effectively individuals who wouldn't have survived, and we're trying to put them into areas where they might survive. I mean, after about 12 months, so this is a active restoration plot in the north end of Port Phillip Bay, you get a response. It looks pretty good. And some of this is just recovered on its own, but there's a smattering of Aclonia plants in here at higher densities than we are getting in the absence of active restoration. So it's encouraging. But this is a lot of work, and simply smashing urchins is expensive. Can we do something outside of marine parks? Because ultimately, most of the urchins are actually outside of the marine parks, not inside of the marine parks. And so this is just the tip of the iceberg. The rest of the iceberg is somewhere else. And this is where we've been trying to come up with other solutions to try to drive this. So this is around how can we work with alternative industries that might be able to take an urchin that you pull off a reef, which is not sellable, and turn it into something that you can sell. So the idea here then is, can we row enhance urchins from barrens to turn them into something we could sell? And can we do that cheaply enough that you can actually make money? And if you can make money on it, then that profit can actually go back potentially to actually fuel restoration activity. So we've spent a fair amount of time figuring out how do you collect these urchins so they normally just get collected to go straight to market. So how do you collect them so they don't die? What kind of rearing environment in a land-based aquaculture facility do you need? What do you feed them? And how quickly can you actually get egg grade quality roe? And what price could you potentially get for that? And develop a business model to actually potentially drive this forward. So this is where we're currently at. So this is a partnership also with the Nature Conservancy and researchers, but now we've actually partnered with both a national and international aquaculture companies working with Invest Victoria to try to drive the development of a new industry that we can actually potentially then accelerate, in this case, be on this orange curve instead of just the green curve, because we're being smarter about how we actually fund the restoration activity. Okay, and now I want to talk a little bit about the why. So that was a little bit about the how. So why are we restoring marine habitats? And I think most of you think, well, this is kind of a straightforward question, that we have habitats have intrinsic value. They have intrinsic ecological values. They support a whole range of biodiversity, and without those habitats, we don't have it. And that's why we restore habitats. And that's certainly why we've been restoring habitats historically for a very long time. But habitats provide ecosystem services that we care about, and we may not actually restore habitats the same way depending on what we are restoring them for or why we're restoring them. So how do we restore coastal dune vegetation so that they provide coastal protection benefits? How do we restore shellfish reefs to maximize the water filtration benefits that they provide? How do we restore reefs so that we maximize fishery productivity? How do we restore wetlands to maximize carbon sequestration? And these may be exactly the same way you would restore them, just to restore the ecological values, but they might be different. And in fact, sometimes they really are pretty different. So this is a shellfish reef we've been involved in putting in Victoria. The primary aim of this is not biodiversity, it's coastal protection. It's trying to save about 12 homes from falling into the bay. This is a shellfish restoration project in Hong Kong that's aimed at improving water quality. We often restore or create artificial reefs to provide a fishery benefit. It doesn't look like a natural reef in any way whatsoever. And we plant mangroves in ways that are not about how a natural forest would be regenerated, simply to maximize carbon sequestration benefits. What this means is that we actually, even restoration is creating novel environments, even when we're actually trying to be positive about what we do. But we also change the environment in lots of different ways that are also creating novel environments that aren't necessarily intended to be positive. So we harden shorelines, we transfer new species into new environments, we change the climate, we pollute coastal waters, we have extractive practices that can be damaging in many ways. And the scale at which this is happening is huge, massive. We are creating lots of environmental change. And the question is how do animals respond to that change? And sometimes animals respond adaptively. They're smart. Some animals are really good at living in cities. I'm sure many of you could name many that are doing quite well in Perth, for example. But from a marine example, here's an example from cetaceans or whales, where anthropogenic noise pollution is masking whale calls, but whales adaptively change their call volume in order to be heard above the noise. There are also lots of examples of animals that fail to adapt. So they mistake prey, they mistake objects as mates, they mistake surfaces as habitats. And so these are ones where they are getting sensory information that is incorrect about what they are choosing to try to feed on or what sorts of habitats they're trying to associate with. So sometimes animals like bad habitats. And a good example of this is actual agricultural landscapes where birds see this huge field full of great habitat to form nests, but they don't actually know or recognize or have awareness of the risks that humans pose in terms of crop harvesting. So they choose to colonize and nest and breed in a field that gets harvested and they die. And there's actually lots of evidence of this actually happening, as animals can't perceive the risks associated with agricultural landscapes. So this is called an ecological trap. It's when animals make maladaptive habitat selection decisions. So in this figure here, what we have is the fitness value of a habitat. So you can imagine this is a poor quality habitat, this is a really high quality habitat. And animals will have preferences. They like certain things and dislike other things. And the expectation is that if animals are optimal or they're adaptive, there should be a positive relationship here. You should prefer habitats that are good, and you should select again habitats that are poor. Ecological traps form when animals are either attracted to habitats that are actually poor quality, or habitats themselves are degraded, but animals do not perceive that degradation, or it could be a combination of both. Okay, so there's a decoupling of the cue and the fitness return of those environments that animals cannot perceive. So how does the restoration come into this picture? As I alluded to, restoration can create novel habitats. So some of these are extreme. This is actually a conceptual vision. But these are all examples of ones in terms of coral restoration projects, oyster reef restoration, or mangrove restoration. And they're obviously fairly unique, and these are novel environments. And the question is how do animals respond, and do these novel restoration habitats actually provide appropriate resources that animals need? So in some instances, restoration can actually form ecological traps, and we've actually documented evidence, well, other people have documented evidence of restoration actually leading to unintended ecological surprises. Sometimes that's because restoration actually fails to consider what animals need, and so don't actually improve the fitness return of that habitat, but it looks really attractive. So it's like you planted a lot of trees, and all of a sudden there's like a giant forest, and the birds go, woohoo, this is great, and there's nothing to eat. There's no holes in the trees for nesting sites because they're all 10 years old. There aren't any old trees. The other way that restoration can lead to an ecological trap is what's called a perceptual trap. So we might actually be great at restoring the habitat, but really poor at restoring the cues that animals use to select habitat. So an example of this are lots of animals actually respond to the presence of conspecifics, so members of their own species. And those species provide cues, whether they're visual or auditory or olfactory. And when those cues, like someone's got to get there first. So if there's nobody there, there's no cue. And so the habitat might be great, but they don't sense conspecifics. And often the presence of adults is a very good, honest signal of high quality habitat. And when it's not there, they don't colonize. And so there's actually been some really interesting work where you can use audio playback. You can actually, for example, even in a reef context, you can actually play reef sounds and stimulate colonization of those reefs to accelerate biodiversity recovery. What this means then is that if we want to improve restoration outcomes for animals, we need to know more about their behavior, and we need to know more about their ecology. So we need to know what cues animals use to select habitats, and we need to be better about understanding whether our restoration activities are providing the resources that animals need to survive, grow, and reproduce. Unfortunately, we're not collecting the right kind of information to evaluate this. So this is a review that I was a part of where we analyzed all restoration studies and looked to see what do they actually measure in terms of restoration studies where they measured animal responses to restoration. Almost all of them use indirect measures of the fitness value of the habitat. So that's basically you go and say, well, there's 20 of them over there and there's 10 of them over there, so that's a better place because I saw more there. The reason why that can be a challenge is that it doesn't measure turnover. So there could be 20 there now, but you don't know how you got those 20. There could have been 100 there and 80 of them died. And there could be 10 there, 10 went there and they all survived. And so your estimate actually of the fitness consequences are wrong or misleading simply by counting numbers. So it's an indirect measure. It's not always misleading, but it can be. Very few actually measure fitness directly. Almost none of them actually measure anything about behavior, and very few studies actually measure both. If we put this in the context of marine habitat restoration, it's actually not a lot, so you'll see the number of studies are quite low. And actually, we're not very good at actually looking at this across marine ecosystems in general. Most of the studies are from revegetation projects, a bit of oyster reef, a bit of artificial reef, and I think one other. And it doesn't matter what taxa we're looking at. Again, we're mostly looking at indirect measures. We're not actually measuring directly, regardless of what we're studying or what our taxa is that we care about. So ultimately, we're just not collecting the right information to know if we're doing a good job of restoring habitats for animals. And I'm going to give you an example of this, of why this is important. And I've been kind of championing this for a long time, and hopefully I'll convince some of you that this is important. I'm going to put it in the context of artificial reefs. And some of you might think, like, why are we talking about habitat restoration and artificial reefs? I think it's important to put it in the context that sometimes systems really are substrate limited, and actually providing more substrate, even if it's artificial, could actually help in terms of biodiversity recovery. And I'll give you some data to support this. So this is a recent review out of Dave Bellwood's lab, where they actually looked at the correlation between fish abundance, fish biomass, and fish richness as a function of live coral cover. And what this graph shows is that basically the strength of the relationship between these fish metrics and coral cover is actually pretty low. So a lot of fish, it doesn't really matter if it's live coral, they need structure. They're just basically after structure. This is not true for all species, but as an assemblage level, the strength of these correlations are all pretty weak. So an argument could be made that many of these species might actually benefit by simply putting out more structure. So we did some work on this about what are artificial reefs doing? Can they be used as a conservation tool, or are they actually potentially functioning as ecological traps? So we did this with a bunch of different species, but I'm going to give you an example of one small temperate reef fish that my group has done a fair amount of work on. And the first thing was to say, like, okay, well, if you give these fish a choice, you can give them a natural reef, you can give them a reef ball reef, which is, at least in Victorian waters, is the dominant artificial reef that's been installed in nearshore shallow water. Or we said, well, we're better at building reefs, so we're going to build our own, and we're going to give them that one, and we're going to see what happens. So we did that, and wow, they love reef ball reefs. Really like them. So way more proportion of fish chose reef ball reefs when given a choice than these other two. Problem is that was a maladaptive habitat selection decision, because we tagged hula fish and released them on reefs actually in situ, so actually in the bay, and did a mark reciting study, and they die 9 times higher rate if you settle on a reef ball reef than if you settle on a natural reef or one of these custom built reefs. So at least in this case, maybe if people who work on artificial reefs probably would say, duh, reef ball reefs actually are pretty shit reefs. I must admit, I wouldn't want to spend any time on there, but at least it was an example to say, look, let's think about this more broadly. We can get bad outcomes by bad design, and we can be a lot smarter about how we design structures so that we can actually not just be a fish aggregating device, but we can actually improve production of these artificial structures so that we're providing the resources that all species need, but also all life histories need. So, you know, fish recruit, they're teeny. Now, their resource requirements and their shelter requirements are a lot different than a full-grown fish. Okay, so I'm going to conclude. So I hopefully I haven't used up too much time, because I really want to open this up to the panel discussion, which I'm really looking forward to. But I wanted to leave with kind of 3 concluding remarks, which we might come back to in the discussion, we might not. And I just wanted to use this as a backdrop from a paper by Macaulay et al., which kind of puts the sort of the human lens on oceans on a time scale of, you know, what it used to look like before we really modified it, our history of modification, and what this future might look like. And I guess it's putting it in the context of, I guess I want to put it forward about how do we reimagine how we can live with nature, right? So it's not well, we do this here, like we fish here, we conserve here, we produce food here, We need to think about this in a more holistic fashion. Okay, so the first point I want to make is conserving what we have is way more effective than restoring what we lost. And I will give you an example, which I already give you the data, but I'm going to put it in the context. Urchin management. I know I only have to keep urchins at a density of about 10 per square meter if I want to maintain what I have. If I want to get back what I lost, I have to fish them down to 3. More money, harder, a lot cheaper to maintain what I have than to get back what I lost. And this is true. Anybody who's in the space will, I'm sure, agree with that. I think it's also really important as we have more and more restoration research and more restoration practitioners that we are good at communicating to government and the population about being realistic about what we can achieve and over what time scales those will be achievable. We can potentially get vegetated reefs back in about 2 years, whether we've restored the ecosystem services we want them to provide, who knows? Some of the work that John's done, you guys can get seagrass beds back. How long is it going to take to actually get carbon sequestration back? A hell of a lot longer. So we need to be communicating that so people have realistic expectations about what we can achieve. And the last one is the point I've just kind of already made, is that we need to learn, or we need to relearn, how do we live with nature. Anyway, thank you very much for your time.